Alright folks, you are on the last part of chapter 4. This is 4-8 to 4-11. It's the muscle tissue and the nervous tissue.
Muscle tissue is widely distributed throughout the body. The primary function of muscle tissue is to produce movement of the skeleton or other tissues of the body. This results in body or organ movement, maintenance of posture, communication such as facial expressions and vocalization, control of body openings, and heat production. Muscle tissue is composed of cells called muscle fibers. Each fiber contains contractile proteins that contribute to the properties of muscle tissue. These include excitability, conductivity, contractility, elasticity, and extensibility. There are 3 types of muscle tissue: skeletal, cardiac, and smooth. Skeletal muscle is under voluntary control and is usually attached to bones via tendons or other connective tissues. Skeletal muscle fibers, or cells, are long, thin, and cylindrical. Each fiber has multiple oval-shaped nuclei located just deep to the sarcolemma, the plasma membrane of the muscle cell. Skeletal muscle fibers are surrounded by a delicate layer of connective tissue called the endomysium. Multiple muscle fibers are bundled together to form fascicles, which are covered by a dense connective tissue layer called the perimysium. The perimysium contains the nerves and blood vessels that supply the muscle fibers. Numerous fascicles are then collected to form a muscle that is covered by an external sheath of dense connective tissue known as the epimysium. The cytoplasm of a skeletal muscle fiber is called sarcoplasm. The sarcoplasm of a muscle fiber contains long cylindrical structures called myofibrils. Each myofibril is composed of bundles of short myofilaments. There are 2 types of myofilaments: thin filaments, which are composed primarily of the proteins actin, tropomyosin, and troponin, and thick filaments, which are formed by the protein myosin. The 2 types of myofilaments are arranged in parallel, and when viewed along their long axis, they exhibit alternating dark and light bands known as striations. The dark bands, called A bands, represent the entire length of the thick filament. Conversely, the light bands, called I bands, represent thin filaments only. The Z discs, dark transverse bands at the center of the I bands, represent fibrous protein structures composed of connectin, to which the thin filaments are anchored. The sarcomere is the functional contractile unit of skeletal muscle. It is defined as the region within a myofibril between adjacent Z discs. There are numerous sarcomeres in each myofibril. Cardiac muscle is found only in the walls of the heart and is under involuntary control. Cardiac muscle fibers are short branch cells with 1 or 2 centrally located nuclei and numerous mitochondria. These fibers exhibit striations and banding patterns similar to those seen in skeletal muscle fibers. Cell junctions, called intercalated discs, are unique to cardiac muscle. 2 specializations within these junctions link cardiac muscle fibers both electrically and mechanically. Gap junctions form an electrical connection by allowing ions to flow from cell to cell, facilitating the spread of impulses that enable cardiac muscle to contract as a unit. Desmosomes contribute to intercalated discs as mechanical junctions that prevent cardiac muscle fibers from pulling apart during contraction. Smooth muscle is located in the wall of blood vessels, ducts, and organs, and is under involuntary control. Smooth muscle cells are elongated, with tapered ends, fusiform-shaped, and contain a single, centrally located nucleus. The myofilaments are not arranged in any specific pattern, and therefore do not produce visible striations or form sarcomeres. Instead of the Z-discs present in skeletal and cardiac muscle, smooth muscle contains dense bodies that serve as attachment sites for thin filaments. Dense bodies are small protein concentrations distributed throughout the cytoplasm. Typically, smooth muscle cells are tightly packed and arranged in sheets. Their contraction is slow and resistant to fatigue. Like cardiac muscle, some smooth muscle fibers contain gap junctions that spread electrical impulses and allow smooth muscle to contract as a coordinated unit.
By the way, she said some of them do, and in fact, the smooth muscle sheets that do have gap junctions here are called unitary or single unit smooth muscles because the whole sheet contracts as one unit. But the smooth muscles that don't have those junctions are called multi-unit smooth muscles. And the muscles in a sheet contract differently. There's certain sections that contract while others don't, and they're under different nervous control. So we'll talk about that when we get to the muscle chapter. So this is the collage of the skeletal muscle, cardiac muscle, and smooth muscle. That video said it all. There's nothing more I could really tell you. All right, nervous tissue. Now this video is going to say it all, so pay attention and play it back as many times as you want. But it's very comprehensive, and there's really nothing else to add to it at the end.
The nervous system is a network of specialized cells that monitors the internal and external environment, processes information, and initiates commands through which the body reacts. Nervous tissue is composed of two types of cells: neurons, or nerve cells, and glial cells. A neuron, the basic functional unit of the nervous system, is specialized to conduct electrical impulses. Neurons have several important functional properties: a high metabolic rate, most are incapable of cell division, and they typically function throughout life. The size and shape of neurons is variable, but they share basic features. The cell body, or soma, receives, integrates, and sends nerve impulses. It typically contains a large nucleus that often has a prominent nucleolus. Ribosomes are abundant and form clusters known as chromatophilic substance, or Nissl bodies, that stain darkly with basic histological dyes. Two types of processes project from the cell body: axons and dendrites. Axons direct information away from the cell body. Most neurons have a single, long, thin axon connected to the cell body at a region known as the axon hillock. The axon generally does not branch, but it may have occasional side branches called collaterals. Most axons terminate as multiple fine branches called telodendria. These have distal expansions known as synaptic knobs, also called end bulbs or terminal boutons. Dendrites direct information toward the cell body. They are relatively short, tapered processes that branch along their length. They often possess small, irregular projections called dendritic spines, where axons can synapse. A synapse is where information is transferred between nerve cells, or to an effector cell, or to a sensory receptor cell. Glial cells, which are also known as neuroglia, are support cells that represent about 60% of cells in the nervous system. Four types of glial cells are found in the central nervous system, or CNS: astrocytes, ependymal cells, microglia, and oligodendrocytes. Two additional types of glial cells are found in the peripheral nervous system, or PNS: Schwann cells and satellite cells. Astrocytes are the most abundant type of glial cell in the CNS. They are star-shaped, with numerous branching processes whose expanded ends contact and support neurons. In some astrocytes, these processes contact capillaries as perivascular feet that help form a blood-brain barrier. This regulates the passage of substances between the CNS and bloodstream. Other important astrocyte functions include regulation of the chemical environment and promotion of neuronal development. Unlike neurons, astrocytes are capable of cell division and can replace dying neurons with a glial scar. Ependymal cells are epithelial cells that line the ventricles of the brain and central canal of the spinal cord. They play an important role in the production and circulation of cerebrospinal fluid, or CSF. Microglia are small elongated cells that have short, irregular processes. Their main function is to respond to infection in the CNS. When activated, they become phagocytic and remove cellular debris associated with damaged nervous tissue. They also secrete immunoregulatory substances, such as cytokines, as part of this response. Oligodendrocytes have large cell bodies with thin, flat processes that wrap repeatedly around portions of axons in the CNS. This wrapping process produces an insulating cover known as the myelin sheath. Each oligodendrocyte can myelinate multiple axons. However, each axon requires multiple oligodendrocytes for complete myelination. Two additional types of glial cells are found only in the PNS: Schwann cells and satellite cells. Schwann cells, also known as neurolemocytes, myelinate PNS axons. Each Schwann cell forms a myelin sheath around a single axon, unlike oligodendrocytes in the CNS that can myelinate multiple axons. As with oligodendrocytes, multiple Schwann cells are required to myelinate each axon. Satellite cells are small and flat. They provide physical and chemical support for neurons located in ganglia.
Okay, folks, so that's your neurons and your glial cells. Make sure you memorize the 4 glial cells of the central nervous system and the 2 glial cells of the peripheral nervous system. Make sure you do that. All right, now we talk about tissue injury and repair and inflammation, and we talk about inflammation several times this semester, and it's always the same thing, by the way. It's not like it's a different thing. So first of all, when you're injured, in A right here, we release some cytokines that make our capillaries permeable or more permeable to what's in the blood. So we have leaky capillaries. So one thing that happens with inflammation is, first of all, we release cytokines. They are prostaglandins, histamines, and kinins. I will show you that in a second. And number 2 is the capillaries become leaky. Now that leads to all the symptoms, signs and symptoms of inflammation. You're going to say, well, what is inflammation? It's swelling, redness, heat, pain. That's what inflammation is. Well, the leaky capillaries account for 3 of them anyway. Why does the area swell? Because blood's leaking into it. Why is it red? Because blood is red. Why is it hot? Because blood has core body temperature versus your skin temperature. Now the other thing is, why is it painful? That has to do with one of the cytokines. There's 3 cytokines: prostaglandins, histamines, and kinins, and prostaglandins cause you pain. So anyway, I have the first step here is injury, and I have leaky capillaries. When the capillaries become leaky, macrophages migrate to the area. There's chemotaxis going on here. Chemicals are calling macrophages to the area. These macrophages will fight off any bacteria that get in through the cut. And if there's any debris, like, say, wood from a sliver, the macrophages will also chew up that. So they'll remove any debris and infection from the site. Now, as soon as the macrophages do their cleaning, fibroblasts move into the area. And fibroblasts put down connective tissue right here. And it's called a granulation tissue. Now, hopefully the granulation tissue is replaced by normal tissue later. So if this is not a horrible cut, then normal epidermal tissue in this case can replace that granulation tissue. The granulation tissue just seals it up for right now. But if this is a bad cut, such that the normal tissue can't replace the gap. Then granulation tissue replaces the normal tissue, and that's a scar. So if the granulation tissue persists, you have a scar. If the granulation tissue is replaced, in this case by normal dermis and normal epidermis, then you don't have a scar. So that's tissue injury and repair. Here's the inflammatory response.
The inflammatory response is an important nonspecific defense against tissue damage. It begins when injured tissue cells release chemical signals that activate the endothelial cells of nearby capillaries. Within the capillaries, adhesion molecules called selectins are displayed on the activated endothelial cells. These adhesion molecules attract neutrophils, slow them down, and cause the neutrophils to roll along the endothelium. As the neutrophils roll along the endothelium, they encounter chemicals that activate integrins, which are adhesion receptors on their surfaces. These integrins then tightly attach to adhesion receptor molecules on the endothelial cells. This causes the neutrophils to stick to the endothelium and stop rolling. This accumulation of neutrophils along the walls of the capillary is referred to as margination. The inflammatory mediators released by the injured tissue bring about changes in the environment that cause mast cells to degranulate and release histamine. Histamine causes vasodilation and an opening of the junctions between the endothelial cells, allowing fluid and leukocytes to leave the capillary and enter the infected tissue. The neutrophils now undergo dramatic changes in shape and squeeze through the endothelial wall into the interstitial tissue fluid. This process is called extravasation. The neutrophils, followed by other types of phagocytes, are attracted to the damaged site by chemotactic substances released by bacteria and tissue breakdown products. They ingest and destroy invading bacteria.
Okay, one last thing before I move on. That's a pretty good video in itself. There's another word for this extravasation. By the way, if you look at the word, where the heck is it? Right there. If you look at the word extravasa, that's outside the blood vessel. Vasa means blood vessel. But there's another word which means the same thing, and that's diapedesis. And I've heard diapedesis. And I've actually heard diapedesis used more clinically than extravasation. I've heard them both used, but I've heard diapedesis used more. So it's when the white blood cell crawls out of the bloodstream. Diapedesis or extravasation. All right, this picture is not in your book. It just tells you the three signs of inflammation: heat, redness, pain, and swelling. There's a lot of debate on whether inflammation is good or bad. Sometimes it seems like it's good because it draws white blood cells to the area and that hastens the healing process. But you know, when you're so swollen that you have loss of function, it's not necessarily good. So I would say this: if the inflammation is not severe, if it's in moderation, then it's probably a good thing because it is going to hasten repair and healing. The three main chemicals in inflammation are prostaglandins, histamines, and kinins. There's some other ones, as you can see here, but those are the three main ones: prostaglandins, histamines, and kinins. And they lead to all the things you see here: leaky capillaries, which lead to heat, redness, and swelling. Prostaglandins lead to pain, so you can see that. So that's injury and repair. I think this is the picture from your book. It says the same darn thing, that you have damage, then you have vasodilation, and then the blood leaks and white blood cells leak out as well. They fight away the toxins in the waste, and then you heal. So that's basically the tissue injury and repair. Now with aging, a couple things happen with aging. Aging is always the last part of every chapter. So what happens when you get older? Well, for one thing that happens is your tissue repair decreases. That's one thing. That could be due to lower levels of hormones. It could be due to a lot of things, but your tissue repair slows as you age. Another thing is that your your stem cells in your tissues, like the stratum germinativum of your skin and other stem cells in your tissues, they only have a finite number of divisions that they do. So if you live to be 200 years old, chances are you're going to have big tissue problems because your epithelial tissue is going to be incredibly thin, your connective tissue is going to be just about gone, et cetera, because these cells, fibroblasts and epithelial stem cells, they only have a finite number of divisions in them. So that's enough to make us last, 100, 110 years pretty decently. You don't see 105-year-olders walking around with no skin, but you see them walking around with very thin skin. So some things happen when you age, and that is the tissue doesn't repair or replace itself as readily. Another thing that happens in tissues is this: uncontrolled mitosis. If you don't die of something else first, that means getting hit by a bus or drowning in Lake Ontario, whatever. I mean, I know it's horrible to talk about death, but if you don't die of something else first, you will get cancer. Now, I'm not saying cancer is going to kill you because we're getting pretty good at treating it. But if you live long enough, you will get cancer. And I think in your lifetime, if you're young, in your lifetime, we'll cure cancer. I think we will. We'll see. I think we're getting close. And that's just because eventually your tissues and cells lose control over mitosis. That's why eventually they're going to lose positive control over the cell division cycle. Cancer is the number 2 killer in the United States. Acute myocardial infarctions is number 1. Heart attacks. So heart attacks number 1, cancer number 2. And that's it, folks. See you in the skin chapter.